can either provide it directly to me, or if you're not comfortable with that for whatever reason, then your TAs. And please interrupt as I'm going through, because I want to make sure that this is as understandable as it can be, and whether it's too simple or too complicated, too fast or slow, feel free to jump in. I don't know on the timing, because again, first time I'm giving this lecture. There are enough slides, hopefully, to get us through the time. If we go quick, great. If we don't get to the last two topics, that's fine too. I'll figure out how to work them in for another lecture some other time. Okay, so I was asked, usually I give the GWAS and linkage lectures. I've been doing that for a long time. Those now, other people are now doing those, which is great. I may try to save, if there's anything specific, GWAS or linkage you have questions on, if we have time at the end, we can come back to them. But I was specifically asked to talk about advanced topics. So I think these are some advanced topics that I think people need to know about. These are things you're going to read about. These are things that you're hopefully going to be thinking about or using in your own research. So Mendelian randomization, pretty popular topic these days. Probably less aware of collider bias and how it can impact not just Mendelian randomization studies or studies in general. And then if we get to it, some rare variance analyses. I don't find these super fascinating. I think we still don't do a good job of it. There's some standard approaches. They haven't changed all that much, but it's something probably we should go over if we have time. Okay, here's the outline. Don't worry about this. This is for when I share the slides around. These are the things we're hopefully going to cover. You don't need to focus on that at the moment. Okay, the question that we want to address is how do we assess a causal observational relationship in the presence of potential confounding factors, okay? So we have this, where we have higher exposure is dietary calcium intake. We see that there is an association with decreased levels of femoral fractures. So this is observed from data. We can go out and collect a big sample. We can say, wow, we see this association. Is it causal? And the reason I might want to know is like, oh, should I tell people who are at risk of fractures, you should take more calcium in your diet? Right, so and you can think about this in any drug. If I have some observed relationship, should I go ahead and prescribe a drug, or should I go and spend $1 billion doing some sort of study that asks the question, right? We can do something like a randomized clinical control trial. The word we have, though, is there could be some confounder, right? It could be something like socioeconomic status. Certain people are more likely to have higher dietary calcium intake, and for whatever reasons, again, socioeconomic status is usually a good example for this, they might have lower levels of femoral fractures, right? So if this is true, where this relationship is induced simply by the confounding factor, we're not going to tell people that they should go take more calcium. We're not going to try to develop drugs that are going along this path. We can save lots of money and lots of people's time. As I briefly mentioned, the gold standard for this would be a randomized clinical controlled trial. We take an appropriately randomized set of people. This is a super important step because if you mess up your first step, then everything downstream is not going to work very well. We can allocate them to an intervention. This might be increase your dietary calcium, and an intervention would be, don't do anything different. And then we can compare the outcomes in these two different groups. We can now randomize, and then we can say what's the odds ratio of having a femoral fracture between these two different groups, and we can get a true sort of assessment of a causal relationship. Limitations, what if we don't have unlimited resources to conduct a trial, right? So collecting samples, doing all this stuff is a lot of work, it's very expensive. What if we have long follow-up times? Think about something like where we want to understand a causal relationship between some factor and Alzheimer's, or something that occurs much later in life. Do we really want to follow people along for 10, 20 years? How big a sample do we have to collect, right? If it's a relatively rare event, we may have to collect hundreds of thousands of people to do an appropriately powered study. And what about phenotypes we cannot randomize? Say the impact of height on cardiovascular disease. It's sort of hard to randomize there, right? Okay, so luckily genetics comes into play. And we have these things called instrumental variables. This was borrowed from epidemiology. This sort of had existed in epidemiology a long time ago, and then someone realized, oh, we can do this with genetics. This is really cool. So you can imagine if we have some sort of instrumental variable, and here it's going to be comprised of genetic variants, and it's causally associated with our exposure. We can then test the relationship of our instrumental variable on our outcome, right? So we have a proxy for this. If this proxy is causal, then we're good. And if it's randomized, we're good. And so when we think about genetics, right, that's where Mendelian randomization comes into the title, right? We think that these are randomized at conception, right? So random segregation of alleles. If that's violated, we have some things to worry about, but hopefully in most cases when we do our studies, those are randomized. Okay, so there's some really key assumptions for this to be valid. So one is relevance. The instrumental variable is associated with the exposure, that's this. If this is not true, clearly this whole method fails. It has to be effective random assignment, which means that the instrument variable is independent of the unmeasured or measured confounders. So that's this sort of thing here, that confounding cannot affect our instrument variable, so there's some things we may have to worry about in the context of even Mendelian randomization with ancestry and things like that. And then the third assumption is what's called the exclusion restriction, and that's the instrument variable cannot have any direct effect on the outcome that's not mediated through the exposure, okay? Because if this is true, we can't really say much about here in this simple framework. Does that make sense? Okay, so keep these three in mind. We'll come back to them, but these are sort of the critical assumptions. When they're violated, our framework falls apart. I'm going to talk about some ways to maybe relax some of these criteria or to detect violation from these assumptions that will allow us to maybe more generalize this approach. So if we think about Mendelian randomization as a form of randomized clinical controlled trial, the randomization occurs at conception. We then have an allocation, which is similar, where we have a genetic predisposition to have increased dietary calcium intake and a genetic predisposition to normal dietary calcium intake. We can also think about, instead of two groups, this could be a continuous measure, right, of calcium intake. And then we can do the same sort of study. So the advantage here is we don't actually need to treat anybody. We can do this in lots of different populations where we have genetic data, right? All we really need is to have that outcome data. So we can potentially save a lot of money or do this in existing studies. That's all clear to everyone? Great. Okay, so the next piece to talk about, because I want to think about this in a practical sense, how will we actually do this, or how do we actually do this? The first thing to really think about is, what are your data sources? And so this is sort of informed by, there's this idea of what's called standard Mendelian randomization, or one sample Mendelian randomization, or two sample. And it's relatively simple. One sample Mendelian randomization, or MR, is occurring in a single study population. So you identify the genetic variants associated with exposure in the study population and their effect sizes, which are their beta estimates. Hopefully you're familiar with that from your GWAS lectures. And then you're going to test the association of those genetic variants with the outcome in the same population, okay? So everything's done, you've collected one sample, presumably you often have individual level data, so you can do the study. And we do have to worry a little bit about confounding false positives, particularly in the presence of weak instrument bias, which I'm going to talk about. So that's a really important factor, is this weak instrument bias. People worry that. So this is where we started. Then a lot of people realized, hey, one way to get around this bias is we can do two sample MR. And actually there's also three sample MR. So two sample MR is relatively straightforward. You're going to identify the genetic variants associated with the exposure in study population 1. You're then going to test for the association of the variants identified in study 1 with the outcome in study 2. So we separated out, or figuring out which are the genetic variants from how we're going to test those genetic variants, right? So we're sort of separating these out. And you can even do three sample MR. One of the concerns is that when you do your initial GWAS to find these variants, you suffer what's called winner's curse. So things that are randomly higher have a larger effect than they, let's say, do in a infinitely sample size population. They randomly rise to the top. So imagine in the true estimate is just below your genome-wide significant cutoff. You wouldn't detect it. But by random chance, it's gone up a little bit. Now you call that a true hit. So it turns out that if you have a finite sample size, that can occur by random chance, so things tend to have larger effects than the true causal effect. And so then when you go test them in your second population, you've overestimated the effect on the first population. So that's called winner's curse. Everyone familiar with that or understand that idea? Okay. So what you can now do is you can say, let's identify the genetic variants in study population 1. We can now estimate their effect sizes in study population 2. So now we've removed the winner's curse, and then we test them on the outcome in population 3. That's called free sample MR. So if you have enough studies, you can do all this. It's fun. The downside today to doing these studies, I'm not going to really talk about, is overlap. So a lot of these huge GWAS studies are meta-analyses from lots of different cohorts. It's not always clear which cohorts are actually contributing to those. So your amount of potential bias is directly proportional to what percentage your population that you're testing your outcome in is actually in any of these earlier GWASs. And it's not always so easy to figure out, so it's just something to keep in mind. that when you just take from a database somewhere of GWAS results and you have your own population, you may still have some overlap. So there are ways to test for that as well. I'm not going to talk about them, but just something to keep in mind. Weak instrument bias. Okay, bias here means the difference between the average estimated value of a parameter and its true value. And it can go in either direction, right? It's just saying how different are you from what it should be if you actually had, true knowledge of what the association is. One sample on R, if the genetic variant exposure association is weak, and I'll define what we mean by a weak instrument, how the strength of the instrument, then chance correlation between the exposure and the outcome will lead to bias towards the confounding association. So this is what we mean. So right, we have this assumption here that this does not occur, right? So that was one of our basic assumptions, that the confounding variable does not affect our genetic variable, and this is going to be true in an infinitely sampled population. We don't worry about this at all because the mean is 0, so it goes towards 0. But if you have a finite sample size, this is not always going to be 0, it's just going to be an average of 0. So it could be that when you did your sampling, for whatever reason, there's some correlation here. And what we've done is when we think about estimating these effects, we think about these arrows. So normally we're trying to estimate this path here, and these other paths are closed off because of our underlying assumptions. So we can get the causal effect of our exposure and our outcome measured through our genetic variant. If we allow this to be non-zero, we've now opened up another way you could get an association. So now this is true, right? So we actually have this association here, and we can get from here, going along this path to here. So we have an effect now on the outcome through confounding. We also have an effect on the exposure here as well, okay? So these graphs are really, really useful. And actually, you can draw them yourself when you're drawing out your populations of potential confounders. These arrows are really helpful when you think about what path you can go on. That tells you what potential sources of bias or confounding exist. So if all the assumptions are met, our causal estimate is this thing right here, or the effect of our exposure and our outcome, assuming there are no other ways to get from exposure to outcome. And that's just, this is really just the difference between this effect, so the effect of our genetic instrument on our outcome divided by the effect of our instrument on the exposure, right? So when we think about an effect, it's the change in the phenotype divided by the change in the exposure or the change in genotype. That ratio, that's just the effect estimate. If you do a linear regression, that's just the beta estimate from your linear regression, right? Phenotype over genetics. Okay, that ratio. So now, if this delta U here is not 0, now there's a difference between what we see and what is the true causal estimate. And if we take our diagram, we can see that it's actually this U on Y estimate. So this is our change in our phenotype times this effect here. So whatever size this effect is, we're now taking this pathway is right here, is this going here, and we divide that by a change on our exposure, our genotype, which is going to be this BZX plus this thing right here. This thing right here. So we're going here as well as here on our exposure. There are two ways now to get to the exposure. There are two ways to get to the outcome. And our bias is essentially this ratio. Does that make sense to everyone? I see a puzzled look over there. No, this is super clear. Great. If it's not, you guys need to speak up.
So just to repeat back what you're saying to you.
Yes.
You're saying we essentially have two possible ways that this, instrument measurement could be influencing or could get to the exposure.
Well, exposure and the outcome, right?
Exposure and then through the exposure to the outcome.
Yeah.
And you're just saying the bias is essentially whatever the ratio is from the desired path over the other possible path to get there.
Yeah.
Okay.
Right. So what we're doing is when we think about doing these things, you can literally just go along these lines. So normally we're closed that we can only go along this line. So that's the true causal estimate is here. If we open up this pathway, we now have to worry about this, right? From going from Z to U to X. So we've changed our exposure a little bit. And we worry about from Z to U to Y, we've changed our outcome. So both our outcome and our exposure are changed and they're correlated, right? And so we take that into account. So we're going to add this little equation here, which is just those other paths. And what you notice here, if delta U is 0, the bias now is 0 because the numerator here is 0, so that becomes 0. Or if our beta Z on X, which is the effect of our genetic variant on our exposure, our instrument strength, if this is really, really large, it also goes to 0 because if I divide some number by a really, really large number, or a relatively large number, it goes to 0. So strong instrument or large sample population, which gets this closer and closer to 0, means our bias goes to 0. Yes?
Sorry, I'm thinking, where are we getting beta from and how, where are we calculating it?
We're going to talk about the actual calculation in a second, but just think about it as it's a regression coefficient. So if you do a linear regression, these are essentially all linear regressions. It's the estimate you get out from the linear regression. Good question.
When we say strong versus weak, is it the effect size on the exposure, or what
So
is that?
here, when we're talking about beta, right, we're talking about the effect of the instrument on the exposure, this estimate of the effect size. The larger that is relative to this other term here, that makes that go towards 0. Okay, so what is a weak instrument? Because, right, that's the whole point here. You want to know, like, you're doing your study, what does it actually mean? Okay, so I'm not going to go how they got to this math, because I actually didn't look it up. This is just, there's a paper that cites this. With certain condition assumptions, 1 over F, and I'll tell you what F is in a second, is the bias of our instrument estimator over the observational estimator, right? So the bias of our instrument over the total effect. F is the F statistic in regression of the exposure on the genotype. That's that beta estimate, right? So the F is the test statistic for that, okay? So when you run a linear regression, if you run this in R, you run an LM model, one of the things you can get out is this F statistic. And so if we think that the sort of rule of thumb that people use is F of 10 is good enough. And that implies that the bias of the estimator is 10% of the bias of the observational estimator, i.e. you remove 90% of the confounding, 90% of the bias due to confounding, so that's good enough, right? So there's still potentially a little bit there. The larger that F statistic is, you remove more and more and more of the bias. And the way we calculate F is N is your sample size, K is your number of instruments. Imagine using 3 or 4 or 5 SNPs or 100 SNPs. And this is just R squared is the variance explained by the instrument. So again, you run your linear model, it'll tell you how much of the variance you explained, how much of the change in the exposure you explain by using SNPs. So keeping that in mind, if I have lots of poorly associated SNPs, that's bad. So in fact, you can actually gain power. Sometimes you can gain power by dropping weakly associated SNPs. Or obviously, if I increase my sample size, this number gets bigger also, right? So bigger sample size, larger effect variance are going to give you larger F. F of 10 is good enough. The bigger F is, the less bias you introduce. Yes?
From like a biological relevance perspective, are there consequences to dropping the weaker variance to have to like increase power?
I'm going to generally say no. And the reason I'm going to say that is I think the biological relevance is an important question, particularly when you think about pleiotropy. SNPs can do lots of different things. The concern would probably be the other way around, including weak instruments or SNPs that have small effect sizes. They might be acting on the exposure through some other mechanism, right? So you really want to get as close as possible. So maybe a good example would be if you want to look at the expression of a gene and say whether a change in that gene expression is causal for a phenotype. So one thing you might do is I'm going to only take SNPs that are in known regulatory regions sitting right around that gene. So there, the biological relevance is very clear. These are cis eQTLs. They clearly affect gene expression. They're not doing it through some other weird way. And so now you're directly asking the question. Alternatively, I could say, oh, you know what? I want more variance explained, and I'm just going to take genome-wide, that are associated with my gene expression. There, though, they might be acting through completely different pathways. So they're probably going to be weaker associations. They may not really be getting at the causal relationship. So I would say it's sort of probably the other way around. Removing those weak instruments, if you want to ask about causal relationships, is probably a good idea. And we can think about where those weak, I'll come to comment later, I'll try to remember. There is a value for those for other things that we can talk about, particularly polygenic CRISPRs. Okay. So the question is like, how bad is this, right? So you sort of want to know, like, is this really a problem or is this just a statistical argument that doesn't really matter? So there's this nice study done by Stephen Burgess. And you guys, if you ever do Mendelian randomization, there are 2 people you should know, Stephen Burgess and George Davey Smith. Most of my lecture is based around papers that they've published. because they're the two sort of dominant people in the field of Mendelian randomization. Okay, so here's real data. They took a study that has 35,000 people. They then can break it into sub-studies randomly. So taking that same study, you can treat it all as one big study, or we can break it down into 250 smaller studies. And we can say, what's the observational effect? This is CRP on fibrinogen levels. And so this is what, if we just measure those levels in people, you can see that they're highly correlated, right? So that's what we're seeing here. There's a very strong correlation, very small standard errors. It's super significant. You could do the Mendelian randomization, two-stage least squares. I'm going to explain exactly what this means in a second. But these are two sort of different approaches. And here's the F statistic. So in our full-size sample study, it's 152, huge F statistic. We think that should work really, really well. As we split into smaller and smaller samples, that N goes down, so the F statistic value goes down. We get some pretty weak instruments. And you can look what happens to our estimates. We think this is probably the causal estimate, which is not causal at all. And then as you get weaker and weaker instruments, you get closer and closer to this to what you see when you just look at an observational and don't ask about the causal relationship. So that's what the weak instrument bias is. It moves you towards whatever is the observational measure. That's what it moves you towards. And you can see this is a really nice example that if I take an underpowered study, I can like, oh wow, there's this clear causal relationship between CRP and fibrinogen, when in fact, that's not true at all. And if you notice this, the standard error here, this is super significant. But it's a false positive, okay? So due to weak instrument bias, this is what it does in practice. And if you want to do one sample MR and worry about weak instrument bias, great paper to read. It really goes through all the details and everything else you would need to try to do it properly. And you might be thinking like, well, why would I ever do one sample MR? Because clearly there's this weak instrument bias, I wouldn't want to do this. But there are some reasons you might want to do it. So two sample MR requires matching on population characteristics, right? So you can imagine different LD can impact the validity of our instrument variables. Genetic effects can vary with time. So if I have different populations, let's say different ages, I may lose some of my validity. Genetic effects can vary by sex. Exposures may vary by population. Imagine you have a population where you've done your initial first stage, or you're in people who have high levels of smoking, and then you want to ask a question another population. Turns out they have low levels of exposure, which is smoking. Might completely invalidate your study. Or you have to worry about harmonization of SNPs, right? So when you do different studies, people use different platforms. They may impute in different ways. You may have a subset of SNPs in one that are not available in the other. You have to do, in fact, spend a lot of time doing data harmonization. So you might be like, I've got the UK Biobank. I've got 500,000 samples. I don't need another study. I'm going to do it all in my study. So there might be a good reason to do it. So the other advantages, one sample MR, you often are going to have access to individual level data. Presumably you're doing that one sample MR in your own population where you've got everything. So you can include confounders, you can stratify in different groups and say, I want to look at young people, old people, smokers, all these other features. That's our subgroup analysis. And then you can potentially investigate nonlinear effects. I'm going to come back to that. Nonlinear effects are probably a disaster. And I'll explain more. Okay, other issues we worry about, concerns about independence between SNPs. The assumption is they're randomized at conception and traits that they don't directly, and we have to worry about traits they don't directly impact. So what are the things we actually have to worry about? Population substructure, right? So genetic associations due to change in frequency with geography, not causal pathways, are something we have to worry about. So a good example of this is lactase persistence in milk consumption in Northern Europe. It's due to the north-south genetic line, not because there's actually a causal relationship here. Assortative mating, individuals reproduce with people who look like themselves. Genetic effects can represent social differences, not causal effects. So you have to think about when we say that they are random in conception, they're random within some framework, right? So people tend to marry and have kids with people who look like them or are the same ethnic group or cultural group. So we have to think about that and how that might have an impact. Dynastic effects are pretty interesting to think about. So parents' genotype can impact a child's outcome by causal pathway, does not impact the child's phenotype. Can anyone think of any examples of this? All right, that's going to be homework. Come up with an example of a dynastic effect. I can think of a good one. There might be others, but I think that would be a good homework assignment to think about how that can impact your results. Okay, so one potential way to sort of deal with all these things is within family MR. So you could, if you have a large enough population, you could actually do Mendelian randomization within family groups. So actually, if you take a population, I'm gonna pick on the UK Biobank because they do a lot of work there. They've got something like, I want to say 20,000 SID pairs. They've got 4,000 mom kids in there. So you actually, even though they weren't collected for family structure, there's a lot of family-related individuals. So you can think about, approaches of using Mendelian randomization within related individuals to get around some of these concerns. How do you select genetic variants? Going back to your question earlier of should we drop against, are we concerned about dropping weaker associations? You can think about single gene approaches. So here, high specificity, presumably high biological interpretability. You pick the gene because you think this gene is in a causal pathway. You understand that pathway. You're testing a very specific hypothesis. So I think there's a lot of value there if you have good instruments. It does limit some of the robustness tests that I'm going to talk about because in this case, all your instruments are probably going to be valid or invalid, right? So if something goes wrong by picking that gene, it's all wrong. And if they're all right, that's great. But a lot of the tests we have on instrument validity depend on at least some of them being right. So if they're all wrong, you're sort of in trouble. And if the trait is truly polygenic, right, then you've reduced your power potentially, right? So if the causal pathway is represented by lots of genes, lots of different genes are all in the same causal pathway, if I only pick one of the genes, I've lost a lot of power potentially. So I would say more common is a polygenic approach, but this works really well if you want to ask a question about a gene or an EQTL. The polygenic approach, again, you could take a biological approach. You could select variants from multiple genes known to be involved in the pathway. I'd be a little hesitant here because I think our ability to pick genes historically has been terrible. So if you look, let's say, pre-GWAS era, like all these studies we proposed, hey, we're going to do an association between these 10 genes and blood pressure, and we'll find some that are associated. We did a terrible job. It wasn't really until we did unbiased scans that we actually found any real genetic association. So I would probably shy away from that. Maybe we have a lot more knowledge now, we can do better. There's also a statistical approach where we include all variants that meet a certain significance threshold. As I talked about, we worry a little bit about the winner's curse, but we can potentially deal with this with 3-sample Mendelian randomization. And then the other thing we worry about with the polygenic approach is the idea of pleiotropy, which I'm going to talk about. But thinking about that diagram of causal pathways, if we violate our assumptions by pleiotropy, we could potentially run into problems. Okay, so what do we mean by pleiotropy and validity of instrument variables? So here are a whole bunch of different diagrams we can think about how genetic variants act. So at the top, we have genetic variants. They directly affect our exposure. Our exposure does something else. It affects what we'll call a mediator, and then it hits the outcome. Perfectly valid. We're fine with this one. This is still going to tell us that this exposure is causal on the outcome, right? If our genetic variants are, we're fine. It may be something downstream, there's a mediator, but that doesn't change our inference about causality. Does everyone buy that? Okay, and that's what's called vertical pleiotropy. We'll distinguish that from horizontal. You could also have the reverse of that. It could be a genetic variant hits a mediator that we didn't know about, so we're looking at an exposure. It could be, that this is our exposure to blood pressure, this is some other mediator of blood pressure, but it's still going to tell us that there's a causal relationship. We buy that also. You can also have genetic variants have an effect on exposure. That exposure could also do something related to some other function, not related to our outcome. We don't really care. It still tells us that this is a causal pathway, right? There's nothing going outside of it. If we have a genetic variant that hits some related variable that it also affects our exposure, but there's no arrow here, that is not a valid instrument. We've now claimed that this variable is causal, our exposure, we've now claimed our exposure is causal, but in fact it's not. So this would be classic confounding. This is just, you have the wrong instrument variable, there's no validity at all. Horizontal pleiotropy is a little more complicated because our genetic variant is associated with our outcome. It's also associated with our exposure, but it's also affected for our outcome. This is what this arrow is telling you, not just through the exposure. So it's affecting something else that's also affecting our outcome. So now we worry about that a little bit because we're not sure how to interpret that result. And we'll talk about some ways to get around that. And then you have what we worry about also a little bit is potentially reverse causation. The genetic variant affects the outcome, and the outcome affects rather than the exposure. And we'll talk about this in bidirectional Mendelian randomization. We can also ask questions about direction of effect. Is A causal on B, or is B causal on A, or both causal on each other? So it's a nice use of Mendelian randomization to actually answer that question. So our primary analytical approach, so this goes about how we calculate or do a Mendelian randomization. So there's one is called the two-stage least squares. It requires individual level data. So in stage one, the exposure is regressed on genetic variants and any relevant covariates. So stage one, that's why we have the S1. We're going to run a linear model. This is sort of what this might look like in R. And we're going to take all our instrument variables, our series of SNPs that we have in our data, and include typically age, sex, genomic PCs, and technical covariates. You don't really want to include other things in here because now you're potentially regressing out causal pathways. Right, so if I want to test some exposure on an outcome, you don't want to actually remove causal pathways. Thinking about the effect of a genetic variant on diabetes, you may not want to remove out BMI, right? If I put BMI in here, I've now removed one of the causal pathways for that SNP. Now it could be, I am not interested in that causal pathway. I want to find SNPs that are exposures that are causal for type 2 diabetes that are not acting through BMI, so I might regress that out there, but typically you don't want to do that. And the stage two is you take the outcome from here and you regress it on the prediction from here, and you regress it on the outcome. What we're doing here is now, instead of taking the exposure, we have now a genetically predicted exposure, right? So we're now saying, what does that exposure look like if we only think about the effect of SNP on that exposure? So this is a predicted exposure, and we test that on the outcome, okay? So that's this two-stage least squares, and we get out a beta estimate for this effect, and that is our causal estimate. Is that clear to everyone what we're doing here? Taking that as a yes. Is that actually clear? Okay. The other approach that probably is much more common because most people are now using summary level data. The reason you use summary level data is often you want to have as big a possible stage one to find your SNPs and estimate their effects. And so you're using published data from huge consortium now, right? If you wanted to look at height, I don't know, what is it, like 8 million people for height GWAS or something like that now? I mean, it's crazy. So people are typically going to take really, really big GWAS, and then they're going to use that at least as their stage one, and so that's why you're often using summary data. And then the standard approach is called inverse variance weighted method. For a single variant, the causal effect is simply the ratio of the variant outcome association, so that beta zy, so the effect of the SNP on the outcome, divided by the variant exposure, the effect of the SNP on the exposure. So let's say your outcome is diabetes and BMI, it's the effect of the SNP on diabetes divided by the effect of the SNP on BMI. For multiple variants, you just combine weighted by the variance. That's why it's called inverse variance weighted. And so that essentially accounts for the confidence in each individual estimate. You can put them all together. These two approaches, in fact, are equivalent under some assumptions, but generally are equivalent. And it's the most efficient estimate of the causal effect when all instrument variables are actually valid. Okay, so that's the thing to remember. This is the most powerful approach, best approach you can do under that assumption, and we can talk about how we test. We'll talk next about how we test those assumptions. There are a couple, for the inverse variance weighted, there are a couple different ways that you can sort of meta-analyze these things together. The standard approach people use is a fixed effect and an additive model typically, and a fixed effect just means that we expect that each variant will give you the same causal estimate. That's not necessarily true. If they're all valid and the relationships of all variables are linear and homogeneous, then that's true. So under that sort of strong assumption that they're all sort of homogeneous, it's a linear effect, that's true. But that's often not true in practice. So oftentimes there's heterogeneity. We can think about different causal pathways for the same exposure. So the more complex your phenotype, think about something like blood pressure, there are lots of causal pathways to blood pressure. To imagine that they're all going to be the same is sort of silly. Variants that influence traits for a longer time may have stronger proportional association with outcomes. So some things might impact you from childhood all the way through adulthood. Some things may only impact you after menopause. So those will potentially give you different causal estimates because the exposure time is different. Nonlinear effects, and often not all of our instrument variables are actually valid. So the recommendation is really what's called a multiplicative random effects model. So one of the things, the advantages of this multiplicative and random, so random effects just allows the instrument variables to have slightly different effects or different effects, and doesn't make the assumption that they're all going to be the same. So it allows for these things to be true. The multiplicative version just means it doesn't upweight the outliers. So if you have a causal effect that looks different than the others, if you did an additive random effects model, it would give too much weight to those. So the reason I'm highlighting this is non-standard. So when you run your standard meta-analysis package in R, it will do an additive random effects model if you request a random effects model. That's not ideal. So just so you know, when you do it yourself, that's just something to keep in mind. And there's a reason for it. Okay. So what does this actually look like in practice? So here we have, on the x-axis, the genetic association with LDL cholesterol. Each of these dots is a SNP. And the lines are the confidence around the estimate. So here we have the effect estimate of this SNP on LDL, and on the y-axis we have the effect of this SNP on risk. This is a log odds ratio for coronary artery disease. And you can see there's a pretty nice linear relationship. So if I were to draw a regression line through here, I would get my effect estimate per change in LDL. I've increased my risk for coronary disease. That's great. That agrees with observational data. That agrees with PCSK9 inhibitors and statins. So that's a really nice positive control. If you didn't see this, you'd start to be a little bit worried that you're doing something completely wrong, right? As an example, if we look at CRP, which is a measure of inflammation, doesn't look like that at all. And in fact, you see sort of maybe a null and some outliers suggesting there's actually plytrophy. These two variants do not look like these guys here. And this is also true, even though CRP is a good predictor for cardiovascular disease. So if you put in a risk model, it's a good predictor, but it's not causal. Changing your CRP levels directly does not do anything. It's a measure of inflammation. So if I can lower your inflammation, that's good, but lowering your CRP levels doesn't do anything, and this is what this tells us. So a good example of Mendelian randomization telling us two things we know. LDL is causal for CHD, CRP is not, even though it's a great predictor. Any questions on this?
I had a quick question. Actually.
Yeah.
So just like, I mean, I think this sort of captures like the big picture of Mendelian randomization. So if LDL cholesterol is truly, causative for, coronary artery disease, then the genetic association should be pretty much the same, because back on that plot thing, the line goes straight through that intermediate variable to get to the end. Okay.
Yep. So it's just saying, the stronger the more variance my SNP explains of LDL, the more variance of CAD, and it's linear. It doesn't have to be. You can imagine that there's some nonlinear effect of LDL on CAD. It turns out it's relatively linear, and that's true in practice, right? So if I give someone a statin and I lower the LDL, the risk, you've changed the risk linearly by your decrease. So it's nice. LDL is just a great example because we actually have drugs that specifically target LDL and like PCSK9 inhibitors, and they have large effects. So you can actually show that this is in fact true. So that's exactly what it is. Okay, so coming back to this this plot here of our Mendelian randomization and these sort of key points of assumptions, relevance, effective random assignment, and exclusion restriction, we can now think about doing some sort of sensitivity analysis. Remember, we talked about worried about that if these are violated, then we're in trouble. So this one is usually easy, right? If our SNP is not associated with our exposure, that's pretty clear. Relevance is easy to figure out there. You just say that we're doing something wrong. B and C are a little more complicated, and there's some approaches that have been developed to detect those and potentially correct for them. So the idea is what they talk about as robust methods for sensitivity analyses. And so the idea here is, can we provide valid estimates of causal inference under weaker assumptions, right? So on our strong assumptions, we've totally closed off those other pathways. Are there ways we can say, well, maybe if those are mostly closed off, we're okay, right? That we don't have to completely close them off. Most methods are geared toward addressing instrument variable invalidity due to pleiotropy and linkage to disequilibrium, not population structure, sort of mating or dynastic effects. And if all variants are impacted in a systematic way, which those things often lead to, it's really hard to detect, right? So we can sort of detect when some things are off. If everything's off in a uniform way, that is much more challenging. From this paper, here's a table. I'm going to talk about some of these methods, but just so you have it in the slides, there are lots of different approaches. There's still probably a reasonable debate about which ones you should do, and there's sort of recommendations. One of the more popular ones is called MR-EGR, and we can substitute the exclusion restriction, right, which is this thing here. So we've closed off that the exclusion just means that our instrument only acts our exposure on our outcome. In fact, what if we allow this to sort of maybe not entirely be true? And we can have a weaker assumption, and that's called the instrument strength is independent of the direct effect, or the inside assumption. I think that's meant to be descriptive. It probably is. It never really makes so much sense to me just reading about it. So I'm going to explain it in a little more detail. I think on the next slide. Okay, so actually I think I'm supposed to explain on this slide. Okay, so the idea here. The idea here is, instead of this being 0, which is normally our assumption, right, that there is no effect here, we can allow there to be an effect going through there. And it turns out, if the strength of the association here is uncorrelated with the strength of the association here, then in fact, the EGR regression, which is a method that's been used for other things, can be used to obtain unbiased causal estimates. The key is, these cannot be correlated. So if they're correlated, imagine that the stronger the effect on the exposure is, the stronger this sort of this is, then the EGR is not going to work, because what it does is makes the assumption that the average is going to be 0. So it doesn't have to be actually 0, but that's the overall is 0. So what does this mean in practice? So the way to think about it is the stronger instrumental variables are expected to be closer to the true causal estimate. So that's this sort of point here, and I'll explain why that is. And weaker instruments are going to be further away from the true causal estimate. So the true causal estimate is this dotted line here. If we run our typical inverse variance weighted slope, we get this red line, which is clearly not our causal estimate. And what the MR-EGR makes the assumption is that these points, because they're associated not just through our causal effect, they're going to randomly be moved up. And the point is they have to be randomly moved up. If they're not randomly moved up, so imagine that the stronger this is, the bigger the effect, I can't re-estimate the slope. So what it does is, because there's no association between the effect estimate of our exposure and the non-exposure causal pathway, they're randomly moved up, and what you can do is you can fit a line, instead of requiring your y-intercept to be 0, you can allow that to vary. And so this amount here, this variance here, is actually the effect of pleiotropy. I don't know if I explained that super clearly, but the idea is that under this assumption, all the points sort of move up. So if I allow my line to no longer have to go through 0, I've now gotten the correct estimate, right? So the beta estimate is going to be correct, even though my y-intercept is not 0. Yes?
So, like, to kind of say it another way, you're basically saying, even if we had no exposure, given this genetic variant, there's going to be this like baseline impact on outcome, and that's the y-intercept.
Yep.
Going up from 0, and then you can say, okay, well now as we increased exposure, how is that changing?
Yeah, and the way to think about this 1 versus 2, right? If you think about, let's see how we do this. So if we think our outcome is some combination of the effect of our exposure, we'll call the other one our confounder. Right? So this is our horizontal pleiotropy effect. And these two things are uncorrelated to each other. What that typically means is that the larger this is, right, so when this increases in value, the overall percentage by the exposure plus the confounder, this value increases. So that means you're getting closer and closer to the true causal estimate, right? Because if this is super large, I almost don't care about the confounder at all. On the flip side, when my exposure effect is small, then the confounder effect is large, so you get further away. Does that make sense? So that's why essentially what you see here is there's a wider spread here, and the further you go, the way it should be tighter. That's what allows us to then shift our y-intercept up and get the correct causal estimate. Does that make sense to people? Seems like Egger regression is great. Maybe we should use this all the time. In fact, there's some problems. The estimates are influenced by outliers. Again, there's some fixes to try to get around this, but that's still a problem. It's prone to be imprecise when instrument variables have similar effect on the exposure, so you're depending on there to being a range of SNP exposures, right? You're trying to fit that line and change the intercept. If they're sort of compressed together, you can't really estimate it very well at all. And it generally has low power. So the way you typically use this in practice, you probably run your inverse variance weighted estimate. You're then going to run MR-Egger as a sensitivity analysis. If the MR-Egger gives you the same result, you're like, phew, this is real. If you get a very different estimate, you'd be concerned and you'd start thinking about what's gone wrong in your study. So it's never going to be your primary analysis because it has low power. It's a sensitivity step. Okay, so what are some other things, alternatives you can take? There's this one called MR-RAPS, Robust Adjusted Profile Score. It first removes strongly pleiotropic variance, then assumes the rest follow the inside assumption. So essentially it's MR-Egger dropping outliers. So that helps somewhat. You can do median Mendelian randomization. You just make the assumption that less than 50% of the variants are invalid. So this is the idea of majority valid assumption. And it's robust to outliers because essentially we're ignoring them. We're just saying what's the median value? So half are above, half are below. So as long as the majority are good, we're good. Okay? There's the mode Mendelian randomization. It assumes that more IVs estimate the true causal effect than any other effect. So that's the idea of the plurality assumption. And the point is there are different assumptions you can make about the data. You can test it in different ways. They should all hopefully give you the same result. When they don't, we start worrying. There's another called MR-Presso. It's similar to the IVW method, but first sequentially remove instrument variables whose causal estimates differ substantially from the others. So essentially you estimate a causal association or causal estimate for each variant. We then say if this one's an outlier, we drop it, we re-estimate, we drop the next one, and you keep going until they sort of look like each other. One problem with that is you may have a clump of causal estimates that are wrong. And so when you're doing a drop one at a time, obviously they're not going to really look like outliers because it's a group. So then there's this constrained maximum likelihood. It tries to select the invalid instruments and estimation, allowing for any of the IV assumptions to be violated. So sort of a more general approach and allows for these clumps of invalid instruments. There are a bunch of papers sort of testing these under different assumptions, things like that. I don't think there's a universal acknowledgement of like, this is the method. They do seem to be powered and work and have false positive negatives under different conditions in different populations. So what do you do with that? You can either just throw up your hands and say, I don't know what to do. There's some recommendations. This comes from, again, there's now a guideline for performing Mendelian randomization. It's updated supposedly at least every 2 years. And so that should really be, I think, the gold standard. It's a fantastic paper. If you're interested in Mendelian randomization, that is the paper to read. Yeah, it's a really good paper. I think very clearly written. So the recommendation, and this makes a lot of sense, is run something like MR-RAPS, weighted median, and MR-CML, as they require different assumptions to be satisfied. That's the inside, or majority, or plurality. So the idea is you're sort of allowing different violations and saying, does my data, you're sort of testing your results under different conditions. If it holds across all conditions, then you're pretty confident in your results. One of the recommendations, explicitly test for heterogeneity in your estimates. So there's a couple different statistical tests to do that. Say, like, maybe I did something wrong about a lot of heterogeneity, that maybe I need to think about how I've selected my variants, my instrument variables. Leave one out analysis, or if you're testing lots of variants, leave a third out. Really testing the idea, your results should be robust to this, right? If I do Mendelian randomization, if I happen to drop 1 or 2 instrument variables, or even 10 or 20 or 30%, I should get the same estimate if it's a true causal estimate. So this is a really nice analysis. If you see something wonky here, something went wrong. Steiger filtering is an interesting idea. This goes back to an earlier comment about causality. It removes variance if their association with the outcome is stronger than the association with the exposure, right? You wouldn't really expect that to be true. If my variant is more strongly associated with the outcome than my exposure, it's probably not acting through my exposure. So it'll show an association, but it's probably not really capturing a causal effect. Probably reverse causation. Okay, so performing two-sample Mendelian randomization, I'm just going to walk through this graphic here. It's from the two-sample MR package in R, and there's another package I'll talk about briefly. But again, just walking us through what we've gotten through so far. We have a bunch of SNPs. They're associated with our exposure. We may have a confounder. Hopefully it's not affecting our SNPs. That's why we use this method. And we want to test it with an outcome. So we do an exposure GWAS. We pick our top hits. We do an outcome GWAS. Then we look up these SNPs in that GWAS. We're not taking these top hits, right? We're looking up our exposure SNPs in our outcome GWAS. That's our second sample. We're then going to be super careful about this to make sure that we've got the SNPs coded the right way. You might worry about AT SNPs and CG SNPs. They're often flipped in the data. So CG looks like GC, AT looks like TA. So you have to do this harmonization, make sure you're in the right units, all the other things you need to do to harmonize your data. This is, I haven't really talked about this, critical step. If you get this wrong, everything is wrong. And then we're going to do this sort of regression analysis. We'll do inverse variance weighting or some other approach. And then we're going to look at the plot of our data. You sort of want to say, does this look roughly right? So sometimes you'll get some result in a p-value, and then you look at it and you go like, that doesn't look linear to me. So clearly I've violated some important assumption. Nice thing about these softwares is you don't really have to pick your method. It does all of them and shows them on a graph. So we've got median, simple median, a weighted median, a penalized weighted median, inverse variance, which is our main one in purple, penalized versions, robust versions, MR-EGR, or whatever, and lots of different approaches. What you'd like to see is they all kind of look the same, right? So that's a nice association. This is LDL. Again, LDL works quite well. This is HDL. So very strong association. Higher LDL appears protective for cardiovascular disease. So when we test it, we put it into risk calculators. It does that wonderfully. But we spent, I don't know, a couple $100 million on drugs to raise HDL. They don't help. And the Mendelian randomization would actually have told you that in the beginning. HDL is not causal. The reason it shows up as an observationally association is HDL is super correlated with LDL and triglyceride. And so what we see here is when we do our IBW approach, which is purple, and some other approaches, we get what looks like a strong causal relationship. And what we, in fact, when we do something like MR-EGR, you can see this is essentially a flat line, right? There's no slope at all. There's no association. So this is a good example of where running multiple different approaches really gives you a lot of insight into the phenotype and the causal relationship. Looking at the individual estimates is important. We talked about this, potentially looking at outliers of pleiotropy. So here we have the causal estimate from each of our SNPs. This one does not seem right to me. I would go and figure out what the heck is going on there, but I would guess that might be acting through some other pathway, right? Super strong association. It's got wide confidence intervals, so probably relatively rare, but that causal estimate is like over 200. Like that seems impossible to me. So that's one where you go back and look to see what's potentially going on there. Another diagnostic, look at the causal estimates in another way is just the different methods. You sort of want to be able to draw a line through these and say like, these all look relatively the same. None of these are significantly different from each other because you've got your agar estimate, IBW, mode-based estimate, contamination mixture is another one I didn't talk about. That, again, a diagnostic, those should all look relatively similar. An additional diagnostic is called a funnel plot, and what we have here is the causal estimate on the x-axis and the precision on the y-axis. And what you expect is a funnel plot, the more precision I have, the closer they should be to the true causal estimate. And the other thing you look for is that they should be symmetric around this. There's no reason they should all be on this side or on that side. That would tell you that there's some sort of pleiotropy driving your association. So you're looking for sort of an even distribution, and they should get tighter as you go on. Yeah?
Are each of these bars one of the different analysis methods?
No, each of these is a SNP. This is a SNP. They're just showing a subset of the SNPs here in this plot. Yeah, good question. And then the leave one out analysis. This isn't, again, I wouldn't do leave one out for something like this because you have so many predictors. I'd leave a third of them out or something like that. But it's a reasonable diagnostic where if you did a leave one out analysis and one of these estimates shifted over when you let that SNP out or went in the other direction, that's telling you there's an outlier driving your result, which obviously we don't want. So this shows that no one SNP is really driving the association, which is important as well. So the two main software packages with tutorials, 2SampleMR and Mendelian randomization, these are R packages. I only put up R packages because that's what I use. They're excellent. So they have really nice tutorials. They're really easy to use. A lot of really useful information. Okay, so we've now talked about sort of basic Mendelian randomization, sensitivity analysis, diagnostics. There's way more you can do with this. This is pretty cool. So people have come up with multivariable Mendelian randomization. Genetic variants are associated with multiple correlated exposures. That was our problem with horizontal pleiotropy, right? So we were worried about this. We can also leverage this. So the idea is if I have genetic variants that are associated with multiple exposures, imagine LDL, HDL triglycerides, and I'm interested in the outcome cardiovascular disease. I was worried if I was only doing one of these, I'm worried about the others. How do I account for them? What this allows us to say, what's the independent effect given the other exposures? So this is where HDL is a perfect example. If I ran it by itself, looks causal, I have to do some other sensitivity analysis to show it's not. I can actually run it all together and I'll find that in fact HDL is not causal, triglycerides and LDL are. So essentially you're jointly modeling the correlated exposures to estimate the independent causal effects for each risk factor. And the only caveat here is if you don't have the exposure measured, right? So if you don't know what it is, then you still have to worry about pleiotropy. But if you are pretty confident that the pleiotropy I'm worried about is through some measured pathways that I know about, this is a really valuable approach. Okay, nonlinear, people have gotten excited about nonlinear Mendelian randomization. The reason they get excited about this is there's various kinds of data that show like J curves, right? So you can, like a classic example is, a classic sort of false example is alcohol consumption and cardiovascular disease or mortality, where people are like, oh, a little alcohol is good for you. And they have this J curve that if people who consume zero alcohol are at higher risk than people who consume a little bit, but if you consume a lot, you're at even higher risk. It's this J curve. And there are other phenotypes as well that this might be true. There's a whole discussion about vitamin D and the benefits of vitamin D, which turn out not to be true again. Oh, if you're really low, maybe there's a benefit, but there's no benefit once you get above a certain threshold. So non-linear effects. So one of the things they looked at in this paper is vitamin D, I think, on morbidity and mortality. It's a large observational data, shows that this is true observationally. So if I look at vitamin D levels of these outcomes, there's a relationship. The initial Mendelian randomization showed no causal associations, but people are like, there has to be something here. So they came up with this nonlinear Mendelian randomization. So what's wrong with this plot? Something fundamentally wrong with this result. This paper has now been retracted because of this. So what they've done, just to clarify, they have an overall effect, which is null. They then break it up into different groups, and they've got their estimates. There's something to look wrong about that. Yeah?
So I could be off here, but my guess is their overall effect is almost exactly the same as sufficient. So do they just have a very, very few samples in all of those other categories?
One guess, but no. Huge study. So it's not a sample size issue.
Is it arbitrary cutoffs?
No. Here's my overall effect. How do I explain my overall effect given these four effects? Can't be, right? If these are all smaller than this, doesn't one of these have to be bigger? Somehow, the overall effect is 0.99, and all the other ones are smaller. Can't be true. If this is true, that this one is really 0.69, some of these have to be bigger to average out to this. So their nonlinear Mendelian randomization is fundamentally wrong. This plot proves that somewhere in their black box software, something went wrong. So in fact, they actually then, this came from George Davy Smith, who's again one of the other giants in the Mendelian randomization field. He pointed this out. They tried another nonlinear approach and this completely goes away. So this is an artifact of the method that they used, and they should have caught it because this can't be true. You don't need to know anything about Mendelian randomization. You don't need to know anything other than a little statistics to know that if overall my estimate is here, if I break it down to subgroups, they have to be around this value on both sides. It can only be all one direction. So nonlinear Mendelian randomization, I would say don't try it at home. It still doesn't work. Okay.
I have a question.
Yeah.
About just MR in general now, its use in the 2020s. Is this a huge, I guess, opportunity for like drug development now? Is this
Oh, yeah.
like the new
Absolutely. There's a whole, I don't have specific, but there's like a whole MR and drugs. But I mean, essentially, it's all the same ideas. Okay, so one other thing they did just to put another nail in the coffin is they used this nonlinear MR approach, NLMR, and they found an impossible association that vitamin D causes age. So they stratified vitamin D into 10 groups. They say, what's the causal effect of age at which you entered the study? Can't be true, right? There's no way that your vitamin D levels determine what age you entered a study. And they can find like, oh wow, we get these really nice association that people with low vitamin D are old, right? Or causally older. Not just that they're observationally older, but causally that makes no sense. So this idea of negative control outcomes is a really powerful approach to any new method or any method in Mendelian randomization. Can't be true. Another really good example of this would be if you were to look at autosomal variants and association with sex. Autosomal variants don't cause sex. Your X and Y chromosome determine your sex. If you found an association between your autosomes and sex, something went wrong. So really nice job of completely trashing the paper and getting it retracted. Okay, so we don't like nonlinear Mendelian randomization. Even the other approaches I think are still not good yet. Not that we shouldn't try. I think it is an important question. But we have to be super careful that anytime you come up with some new method, you have to validate it, and I think validate it positive and negative, right? So we have some very easy things to do. Sex and age should not be causally associated. Very interesting time-varying Mendelian randomization, and this is particularly is called life course MR. It assesses the varying effects of exposure at different periods of your life. A lot of them use childhood and adult BMI is one example of this, where we have measures and those do change over time, and genetic variants are differentially associated with those two things. So we can separate them out genetically, and that's what allows us to do this Mendelian randomization. So this is looking at childhood leptin levels and adult leptin levels. And the idea here for childhood levels, we have our genetic variants that might be associated with both, but the causal effect on childhood leptin levels should be your childhood body size, right? That's our causal pathway. But there could be some correlation here, that's fine. For adult levels, genetic variants affect child body size. That affects adult body size is the idea, and this is our causal pathway. So we have two different causal pathways that we're testing. When we do univariable MR, we find on childhood leptin levels, we find a stronger for childhood body size than for adult body size. And what's interesting to me is that even when I do multivariable, so I'm adjusting for that, it shrinks the effect of adult body size, but not completely. So it does suggest that there's some other intention of it. Yeah. I think what it's telling us, some of these genetic variants are actually, that affect adult body size, are actually violating our MR and are acting through some other pathway that is not childhood body size. So that's what that's telling us. Adult leptin levels, I think, are a little easier to interpret. We look univariable, we see a strong association with those childhood and adult BMI causal associations. But once we do the multivariable MR and we put both in our model, childhood body size is not causal for adulthood leptin levels. That's a really nice example, great. Another example, which I thought was kind of interesting, was breast cancer, and they're actually off the line here. These two values go with these two values. So breast cancer, and this is also body size. So early life body size, adult body size, univariable, protective effect for breast cancer. But when you do the multivariable MR, it's really only child body size that is protective for breast cancer. So I thought that was kind of interesting. where it really is an adult body size profile. This is a causal relationship, right? So we're not asking about observationally, we're asking causally. Genetic variants that predict early body life size are protected, and the ones that affect adult body size are not. Another thing, very cool thing that is relatively new, tissue portioned Mendelian randomization. So the idea is you have genetic variants associated with the trait. Again, we're using BMI and leptin levels. It's from the same paper. And you can actually partition the variants into those that act through adipose tissue versus brain tissue. And the way they do this is they co-localize cis-EQTLs, right? So essentially you do your GWAS for BMI, and we can say what pathways do they act in by asking the question of what genes do they influence. So we know brain expressed genes versus adipose expressed genes. We then do co-localization and say, in fact, these are the same signal. Hopefully you guys covered co-localization. And we can partition them into these two things. Again, univariable MR and multivariable MR, and you can see, in fact, it's the brain tissue that is actually the causal pathway. So genetic variants that act through gene expression in the brain are causal for childhood leptin levels, and this is also true for adults as well. And so when you did the univariable, you would have thought, oh, adipose tissue as well. In fact, that's not the case in the multivariable bit. Adipose tissue is not the relevant feature here. So this is kind of cool. You can imagine using this in lots of other circumstances where anytime you can partition your genetic variants through different causal pathways, we can now use a similar approach. It doesn't have to be tissue, it can be anything, where you can now get fine-grained understanding of causal pathways. All right, is that clear for everybody? Some very useful and new approaches, and you can think about other ways to adapt Mendelian randomization. People are still developing all these new methods. These are all sort of, I'd say, a time where they're really developing a lot of cool new tools. Okay. Another thing people are particularly interested in is the idea of mediation analysis. And this is, there are two different methods. This is called the product method. So in step one, we're going to look at the genetic variants of an exposure. We're going to do our Mendelian randomization on the exposure and then get a causal effects on the mediator. So we've now identified causal effects of the mediator. And then in the second step, we're going to do our MR of genetic predictors of the mediator on the outcome. And then we're asking the question of how is the exposure acting through this mediator? And the way we do that is we take our effect from step one, so the effect of our exposure, causal effect of our exposure on the mediator. We multiply that by the causal effect of the mediator on the outcome. So if we multiply those two things together, we get the indirect effect of the exposure on the outcome. So that tells us how much is going through the mediator. Does that make sense? We just multiply the causal effect here, this causal effect times this causal effect, and now we get the indirect effect of the exposure on the outcome via the mediator. It's relatively straightforward. The other approach is called the difference method, and there we're using multivariable MR. So we do an MR of the exposure on the outcome. We then put the potential mediator in, we do the multivariable MR, and we take the difference between these two, and we say that's the indirect effect of the exposure of the outcome via the mediator. So two different approaches, they get to the same thing, is how much of your overall effect, which is what you get here, is coming through just the mediator. This is the paper that did these simulations. They say the product method works better, and there's this delta method for calculating the standard errors. So, if you wanted to do this, that's probably the way to go. They have some nice results showing, in fact, that that's probably the better approach. Okay, bidirectional Mendelian randomization. We have two different traits. They're correlated with each other, and we want to ask, does A cause B, does B cause A, do they cause each other? So that's what's bidirectional, very simple, and we have a really nice example of that. So the age-old question, does poor eyesight lead to more time spent indoors studying? Or does more time spent studying lead to myopia? Anyone have an opinion which one they think it is?
The latter.
Yeah. I'd buy that that's true. I don't think that like, oh, I don't see, so I'm not going to go play outside. It makes a lot more sense like, oh, I sit in front of my computer all day or I read books all day, I'm going to have bad vision. But we don't know that that's true. So how would you ask this question? We're talking about Mendelian randomization, guys. It answers all questions, causal effects. Pretty straightforward. We take UK Biobank data, we have educational attainment, and we can look at time spent in education, and we can look at your vision. So this is my refractive error. And we can show the more time you spend genetically educating, getting education, is the worse your vision is, right? So this shows a causal effect. We can then look at reverse of that and say, well, does bad vision lead to more time spent studying? No, it doesn't. So whatever, I don't remember which direction this goes in, but whatever it is, it's showing that there's no correlation in the reverse direction. So we've now solved a 400-year-old question, and we can now in fact say, don't study so much, you're going to have bad eyesight. Okay. And you guys really should be thinking, like that last example, you should be thinking about causal effects in the questions you ask. It's really cool. You can ask trivial questions like that, but you still get a nice paper out of it. But thinking about drugs, thinking about other things where you really want to understand causal mechanisms, MR is really a powerful approach if you do it right, right? We've talked about assumptions and biases and all the other things, but if you do it right, it's great. There are ways to do this online. So this is called MR Base. And they have a whole bunch of different catalogs you can choose from to do your Mendelian randomization, methylation QTLs, protein QTLs, the GWAS catalog, different GWAS catalogs. You can do this all online. You go in and you can select your favorite, outcome that you're interested in, coronary disease. You can then select your favorite exposure or risk factor. You then can tell it sort of all these different parameters, how correlated you like, or looking up LD proxies between your two different populations. If I genotype to SNP in one and not in the other, I can say how willing am I to accept a proxy for that variant, minor low frequency threshold. Here are all the different sort of tests you can do, aggregate sensitivity analysis, all the other things, and then it will dump out these nice plots for you and tell you all these nice statistics, right? So I would say this is a place to start. I worry about that, you've got to make sure you do it right, but they try to make it super easy. This is curated. It's a great resource. Okay, so MR is useful if we want to estimate the quantitative causal impact of exposure on the outcome. Again, causal impact. It's not the same question about shared genetic etiology. This goes back to weak instruments and other things like that. If you really want to ask about shared etiology, there are other approaches. I think you guys hopefully have talked about LD score regression. There's also assessing shared heritability with this thing called Rebel, which is a lot of people use for shared heritability. MR is not the most powerful approach to answer these questions. It's specifically about causal estimates. Okay, so keep that in mind. This is not, every tool has a specific use, and using the right tool is really important. Okay, any questions that we've covered in Mendelian randomization? And we still got 15 minutes left for everything else. Yes?
So back to that slide we were just looking at and thinking about the tissue portion of Mendelian randomization.
Yeah.
Sort of my takeaway is look, Mendelian randomization is just a way to use genetics as an instrument to sort of estimate an effect's result on an outcome.
Causal effect.
Causal effect, sorry.
So
Causal effect.
That's the critical piece here. It's getting a causal effect.
A causal effect out of it.
Yeah.
Why do we care which tissues the instrument acts through?
Oh, you'd be super interested. If you thought that the effect of BMI on leptin levels was mediated through adipose tissue, you might think about very different drugs or very different approaches than if I know it's mediated through the brain. If I also want to understand biology, it's super interesting to me. Most people might think, oh, it's all about adipose tissue. In fact, it tells you that leptin is about satiation and gives you some biological insight into what's actually going on. So I think that's sort of the key there when we think about causal pathways and causal mechanisms. So it could be you're thinking about treatment, but even just understand the biology. If I know it's causal this way and this is not causal, it's a secondary effect, I can start thinking about drawing out those diagrams much more complicated, right? And that's where you can spend a lot of time. A affects B, that affects C, and all the other things. The direction of those arrows is super important. Good question. Okay. So I doubt we'll get to rare variant analysis, but we'll at least talk about collider bias. I thought this was super interesting when I read this paper here. So it's really important to think about any of these studies that there is selection bias, okay? So that selection bias is defined as the non-random participation or attrition from a study, right? So longitudinal studies often have attrition. And there are a couple things. There's the healthy volunteer bias. So right, UK Biobank, they sent out, I think 9 million people were asked to be in the participate, only 500,000 participated. So it was not a random subset that participated. As an example, higher genetic risk for schizophrenia is correlated with non-completion of questionnaires. So even if they entered your study, when you're doing a sub-analysis in that relied on a questionnaire, you might find like, oh, there's missing data here. It's not missing at random. A lot of our statistics assume missing at random. And if it's not, that's a problem in here. If you were studying schizophrenia and you were doing something based on questionnaires, you'd actually be missing people maybe with the highest genetic risk for schizophrenia. In this study we're going to talk about, heavier smokers were more likely to not stay in the study. Don't really know why, they just weren't, okay? So generally thought that while this influences the prevalence estimates or outcome of disease, right? So the prevalence of disease might be different if there's healthy volunteer bias or whatever it is. It was not thought to affect association of variables, but it can introduce what's called collider bias. Okay, so collider bias occurs, and I'm going to give you a good example of this on the next slide, when two independent variables, x and y, that are uncorrelated with each other, cause a third variable we'll call z. If we adjust for z when looking at the association of x and y, we'll see a bias in the estimate of the causal association of x and y. So we can introduce a correlation. The correlation doesn't exist if we looked at everyone, no correlation, but because z is correlated with both, if we do something with z, and I'll define what I mean by something, we can introduce a false correlation. Okay? So here's my example. I toss 2 coins. If at least one is heads, I ring a bell. So that one's heads, we ring a bell. That one's heads, we ring a bell. Great. No correlation between them, right? If I toss 2 coins, they're uncorrelated. Okay, so the correlation is 0. Now imagine instead, I hear a bell. I then look at my coin and go, oh, it's a tails. Right? So we've now started with we've selected on hearing a bell. I looked at my coin and I see it's a tails. What do I know about the other coin? Heard a bell ring. It's a heads, right? So now I know that that had to be heads, and now I can do a correlation. If I did this over and over again, I'm going to get a correlation of negative 0.5. But if I see a tails, I know it's going to be a heads on the other side. If I see a heads, I don't know. But I haven't fully captured the effect, but I've gotten a strong correlation. Okay, because I selected based on hearing a bell, I've now introduced a correlation that does not exist it does not actually exist. Okay, so collider bias, we have our genetic variance, we have a phenotype X, we have an outcome in these, notice no arrow between them, they're uncorrelated, but phenotype X and outcome Y were both used to select people into our study. Okay, so now our Z is we're selecting based on something that is separately associated with both. So we've now created a correlation between G and Y because by allowing this to occur, we now have this occurring, right? You can just draw along these lines. If there's a connection, then these are connected. So we've introduced a false correlation, and this is what collider bias is. Okay, does that make sense to people? Because we've selected based on this, we've introduced a correlation between these two things, which also means my genetic variance, which I'm using to estimate the causal effect of this on that, even though there is no causal effect, I've now allowed it to occur. Yes?
I guess I've taken like one kind of like epidemiology course where like they really focused on like the fact that like your attrition or like your like responder and non-responder and like potential attrition group like should not necessarily be tied to like the outcome. So like if that occurs, it's like less of a valid like experimental setup.
Yeah. I'll give you a specific example of that.
Okay.
Yeah. So right, so there is a huge problem if attrition is if there is attrition based on your exposure. There also is what's it called? It's called survival index bias, which I have a reference to. The other thing to think about is when you're selected in, let's say, I'm looking at incidence versus progression of disease. So to see progression, you have to have incidence of disease. And so you get a potential bias there because you've selected based on people having the disease, if they are more likely to be one sex or in age or anything else, you've now introduced a similar sort of bias. So I'll give you an example. Okay, so collider bias, is it a problem in practice? So the ARIES is a longitudinal follow-up of the ALSPAC study. As I said earlier, heavier smokers are less likely to stay in the study, higher educated mothers are more likely to stay in the study, and there's collider bias, and we can go through this. So overall, we're looking at the association between smoking and maternal education. That's what they're testing here. Here's our smoking genetic risk score in the ALSPAC, the parent population, and it's significant. So there is an association between our genetic risk score and whether you actually smoke, which is good. That has to be valid. If we look at who made it into the sub-study, you can see people who smoke were less likely to be in. The smoking genetic risk score had no effect. And maternal education, if you were higher educated, you were more likely to stay in this sub-study, okay? If we now look at the association between smoking and maternal education, in the overall study, smoking, let's see, smoking, yeah. So smoking observationally is associated with people who smoke more have lower maternal education, but the genetic risk score is not associated, which is what we expect, right? So people who are higher educated tend to smoke less, but it's not genetic. It's not a causal association. But if we look at our sub-study, we actually find, in fact, that the genetic risk score is now associated with educational status, suggesting that smoking is somehow causal for educational status, when in fact it's the reverse. People are more educated or less likely to smoke. So because of this bias here, that smoking and maternal education were uncorrelated or not causally correlated, but because there was selection bias based on both features into our sub-study, even though overall there's no causal association, we're now inducing a causal association. Does that make sense? So this is exactly what collider bias is doing. Because we've selected based on two other features, those features we've now introduced a correlation, and when we ask for a causal association, we find one even though in the overall study there isn't. So that's bad. So what are the things you can do to detect or correct collider bias? Use negative controls, which you talked about, age and sex autosomal SNPs should not be associated causally with any of these things. If they are, you should know that there's some sort of collider bias. You can do what's called inverse probability weighting. So instead of just looking at each sample in your selected population, giving them an equal weight in your study, you can say, well, how likely were they to represent the overall population? And so if you weight them by that, in some sense you recreate the overall population and you remove this bias. Compare to other studies, right? So don't just do this in one sample population, do this in another that is likely subject to different biases. So if you see the same result in multiple different studies that have different potentials for collider bias, that's probably good. And there's some variations on the MR-EGR approach. I didn't want to go into them for time, but this paper goes into that in a lot more detail with some nice examples. All right, questions? I think we're not going to talk about rare variant analyses. I hope no one's too disappointed. Unless you guys want to do that in 7 minutes. Okay, questions on what we've talked about so far? All right, what I need to do, which I'm going to put on my list, I'm going to, all the papers are listed here, and the ones you see listed multiple times, you should probably go look at. They're actually really good papers. I found them really interesting and very accessible. So I try to pick papers where they have nice examples, they're not too statistical, so I think I avoided most of the mathy papers. If you really like math, I can give you some really much more mathy papers that explain all this in very, very great detail, mathematically. But I think if you really want to understand this stuff, maybe it's super clear and everything's like, oh, this made perfect sense, this is great. But I would say if you really wanted this to solidify, you should read a couple of those papers. All right, any other questions or comments? Generally, the speed of this was okay, too quick, too slow.
I think it was a bit fast.
All right, I will have to think about cutting out some slides.
I think like the concepts were okay, but I think sometimes like there was a lot of like sub-concepts that I think it was hard, at least for me, to keep track of all the sub-concepts, even if I got like the big picture.
Okay.
I think it would have helped if you had started with an example. So at the very beginning, you were talking about the instrument variables, and I had no clue what you were talking about there. But if you had just said, here's our genetic variants, here's our LDL, and here's our
Okay, good.
cholesterol, or whatever. That would help me.
Okay.
Connect those.
Got that. Anything else? Anything you don't want to share me directly, you can let the TAs know they will anonymously share it. But my goal here is to improve my lectures and make sure you guys understand stuff. So if you have questions, you can always stop by my office, send me emails. But I do really recommend reading some of these papers to solidify this, or even just get onto MR base and like run a couple things. Or the 2-sample MR or the Mendelian randomization package have really nice tutorials, super easy to use. So, and presumably everyone here has done some sort of R. Did you guys have to do some sort of boot camp or something? So if you
Not
haven't,
for R.
huh?
Not for R.
They did Python and Unix.
I don't. There might be Python versions of these, so you can take a look to see if they're Python versions. I mean, I'm not opposed to Python. It's just R has a lot of packages for genetic studies. I think Python's great for doing like neural net stuff and some other things that are better in Python. But for a lot of the genetic stuff, the tools are really written in R. Okay. Thanks, everybody. Yep.
And also, they're having a discussion on Friday, so if you want to,
Yeah. Unless we're asking for not able to come in on Friday.
Okay.
Yeah, I think it's hard to do one person being on Zoom.
Yeah.
But that, I mean, the paper I said is just.
You might ask me.
I think, you know, it's relatively straightforward.
Yeah, it wasn't too long, so.
Yeah.
Oh, boy.
Tell them part.
Oh, no, I don't ever have a finger. I'm wondering,
It's the first time I've really had the opportunity to really dig in.
Yes, we're gonna discuss that. We're waiting on. Yeah, that's.
Sorry.
Yeah,